Today in this video we are going to discuss about the classification of amino acid and its properties. Before going to the classification, we want to know about the amino acid. What are amino acids? Amino acids are organic compounds. They are organic compounds containing amino group, carboxyl group and side chain, that is an R group. Amino acids are organic compounds containing amino group, carboxyl group, side chain R groups. The major key elements in an amino acid are carbon, hydrogen, nitrogen and oxygen. The major key elements in amino acids are carbon, hydrogen, nitrogen and oxygen. About 500 amino acids are known. Only 20 amino acids appear in the genetic code. About 500 amino acids are identified. In this, only 20 appear in the genetic code and the synthesis of the protein. And they can be classified in many ways. So what are amino acids? They are organic compounds containing amino group, carboxyl group and side chain, that is R group. The major key elements in an amino acids are carbon, hydrogen, nitrogen and oxygen, and about 500 amino acids are identified. In this, only 20 appears in the genetic code. Now coming to the basic structure of an amino acid. This is the basic structure of an amino acid. Here you can see a carboxyl group, COOH group here, and an amino group here, then an R group here. So based on the R group, the name of the amino acid is different. So the R group is different in each amino acids. This is the basic structure of an amino acid. Contain amino group and R group, then carboxyl group. So what is the need for classification of amino acid? The classification of amino acids gives the grouping between 20 amino acids and a basic outline for grouping. The classification of amino acids gives the grouping out between 20 acids and a basic outline for grouping. It makes a clear idea to pick the amino acid type. And it is more useful for biochemists for the easy understanding between each amino acids. This classification is more useful for biochemists for easy understanding between each amino acids. These are the need for the classification of amino acids. Now we can see how it is classified. The amino acids are classified based on R groups, or based on the polarity and R group, or based on the distribution in protein, or based on nutritional requirements and the number of amino and carboxylic groups. So the amino acids are classified on different ways. One is based on R group, another one is based on polarity and R group, the next one is based on distribution in protein, and the next classification is based on nutritional requirements, and the next one is based on the number of amino and carboxylic groups. Now, based on the R group, they are again classified into different groups. Based on the R group, they are again classified into different groups. They are first one is simple amino acids. These have no functional group in their side chain. The simple amino acids have no functional group in their side chain. Example is glycine, valine, alanine, leucine and isoleucine. The simple amino acids are glycine, valine, alanine, leucine and isoleucine. They have no functional group in their side chain. The second one is hydroxy amino acids. These have a hydroxyl group in their side chain. Examples are serine and threonine. Hydroxy amino acids contain a hydroxyl group in their side chain. Examples are serine and threonine. Based on the R group, they are again classified sulfur containing amino acids. Sulfur containing amino acids have sulfur in their side chain. Examples are cysteine and methionine. So the next classification is sulfur containing amino acids. The next one is aromatic amino acids. Aromatic amino acids have benzene ring in their side chain. Examples are phenylalanine and tyrosine. Phenylalanine and tyrosine. Next one is heterocyclic amino acids. Heterocyclic amino acids having a side chain ring which possess at least one atom other than carbon. So heterocyclic amino acids having a side chain ring which possess at least one atom other than carbon. Examples are tryptophan, histidine and proline. Tryptophan, histidine and proline. Next classification is amine group containing amino acids. They are derivatives of amino acids. They are derivatives of amino acids in which one of carboxyl group has been transformed into an amide group. One of the carboxyl group is transferred to an amide group. Examples are asparagine and glutamine. So the amine group containing amino acids are the derivatives of amino acids in which one carboxyl group has been transformed into an amide group. The next one is branched chain amino acids. In branched chain amino acids contain aliphatic side chain with a branch. A branched chain amino acid is an amino acid having aliphatic side chain with a branch. Examples are leucine, isoleucine and valine. Examples are leucine, isoleucine and valine. Next one is acidic amino acids. Acidic amino acids have carboxyl group in their side chain. Carboxyl group in their side chain. Examples are aspartic acid and glutamic acid. These are the acidic amino acids. The next one is basic amino acids. Contain amino group in their side chain. Examples are lysine and arginine. So the basic amino acids contain amino group in their side chain and examples are lysine and arginine. The next one is amino acids. Amino acids containing a secondary amino group or amine group is known as amino acids. Amino acid containing a secondary amine group is known as amino acid. Example is proline. So based on the R group, the amino acids are classified into 10. They are simple amino acids, hydroxy amino acids, sulfur containing amino acids, then aromatic amino acids, then heterocyclic amino acids, amino group containing amino acids, branched chain amino acids, and acidic amino acids, basic amino acids and amino acids. Next one is based on the polarity and R group. The classification based on the polarity and R groups. First one is amino acids with the non-polar R group. These amino acids are hydrocarbons in nature. They are hydrocarbons in nature and are hydrophobic and having aliphatic and aromatic groups. Aliphatic R groups are alanine, valine, leucine, isoleucine, proline. And aromatic groups are phenylalanine, tryptophan and methionine. So based on the polarity and R group, the first classification is amino acids with non-polar R groups. Non-polar R groups. These are hydrocarbons in nature and hydrophobic, having aliphatic and aromatic group. So the aliphatic R groups containing amino acids are alanine, valine, leucine, isoleucine, proline, and the aromatic R groups containing amino acids are phenylalanine, tryptophan, methionine. The next one is amino acids with polar but uncharged R groups. Amino acids with polar but uncharged R groups. These amino acids are polar and possess a neutral pH value. These amino acids are polar and possess a neutral pH value. Examples are glycine, serine, threonine, cysteine, tyrosine, glutamine and asparagine. Examples glycine, serine, threonine, cysteine, tyrosine, glutamine and asparagine. Next one is negatively charged amino acids. That is their side chain, that is R group, contain an extra carboxyl group with a dissociable proton. And renders electrochemical behavior to proteins. So it gives the electrochemical behavior to the proteins. Examples are aspartic acid and glutamic acid. Aspartic acid and glutamic acid are negatively charged amino acid because their side chain contain an extra carboxyl group with a dissociable proton. Is positively charged amino acids, that is their side chain contain an extra amino group and rendering basic nature to protein. The side chain contain an extra amino group and rendering basic nature to protein. Such amino acids are known as positively charged amino acid. Examples are lysine, arginine and histidine. Lysine, arginine and histidine. These are the classification based on the polarity and R groups. That is amino acids with non-polar R groups, then amino acids with polar but uncharged R groups, and amino acids having negatively charged amino acids, then positively charged amino acids. Now the standard protein amino acids. The amino acids that are used to form proteins and recognized by ribosome amino acylation systems. Examples are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. So standard protein amino acids are the amino acids that are used to form proteins and are recognized by the ribosome. Examples are histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. Next one is non-standard protein amino acids. They are amino acids, they are not required to build proteins. Amino acids that are not required to build proteins and having a vital role as metabolic intermediates. Non-standard protein amino acids are amino acids that are not required to build proteins and have a vital role as metabolic intermediates. Examples are hydroxyproline, hydroxylysine, carboxyglutamate, diaminopimylate. So examples are hydroxyproline, hydroxylysine, carboxyglutamate and diaminopimylate. Next one is non-standard non-protein amino acids. These are the derivatives of amino acids and have a role in metabolism. Non-standard non-protein amino acids are derivatives of amino acids and have a role in metabolism. Examples are alpha aminobutyrate, citrulline, ornithine and beta-alanine. Alpha aminobutyrate, citrulline, ornithine and beta-alanine. So the classification based on the distribution in protein are standard protein amino acids, non-standard protein amino acids, non-protein amino acids. Now the classification based on nutritional requirements. So based on the nutritional requirements, the amino acids are classified into two. They are essential amino acids and non-essential amino acids. First one is essential amino acids. They cannot be made by the body. Essential amino acids cannot be made by the body because they must come from the food. So they are known as essential amino acids. They cannot be made by the body. And the examples of essential amino acids are arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. These are the examples of essential amino acids. They are coming from the food and not made by the body. The next one is non-essential amino acids. An amino acids that can be made by human and so it is essential to the human diet. They are known as non-essential amino acids. Amino acids that can be made by humans and it is essential to the human diet. Such amino acids are known as non-essential amino acids. The non-essential amino acids are alanine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine and tyrosine. These are the non-essential amino acids. Next one is the classification on the basis of number of amino and carboxyl group. Basis of the number of amino and carboxyl group. First one is monoamino monocarboxylic amino acid. Monoamino monocarboxylic amino acid. It contains only one amino group and one carboxyl group. So monoamino monocarboxylic amino acid. Examples are glycine, alanine, proline, phenylalanine, methionine, serine and threonine. These are the examples of Monoamino, monocarboxylic amino acid, which contain only one amino group and one carboxyl group. Next classification is monoamino dicarboxyl amino acid. Monoamino dicarboxylic amino acid. That means one amino group and two carboxyl group. One amino group and two carboxyl group. Examples are aspartic acid and glutamic acid. Aspartic acid and glutamic acid are the examples of monoamino dicarboxylic amino acid. Next classification is diamino monocarboxylic amino acid. Diamino monocarboxylic amino acid, that is, which contain two amino groups and one carboxyl group. Two amino group and one carboxyl group. Diamino, two amino group and one carboxyl group. Examples are lysine, arginine and histidine. Lysine, arginine and histidine are the examples of diamino monocarboxylic amino acid. Next is the properties of amino acids. First one is physical properties. They are colorless. Amino acids are colorless and they are crystalline in nature. Amino acids are crystalline in nature and the tyrosine is tasteless, but glycine and alanine is sweet in taste. Tyrosine is tasteless, but glycine and alanine are sweet in taste. And they have a melting point above 200 degrees Celsius and soluble in polar solvent and insoluble in non-polar solvents. The amino acids are soluble in polar solvents and insoluble in non-polar solvents. And they have absorbance at 280 nanometer. The amino acids have absorbance at 280 nanometer. So these are the physical properties of amino acids. The amino acids have molecular weight 100 to 50,000 Dalton. Molecular weight is 100 to 50,000 Dalton. All amino acids possess optical isomers. All amino acids possess optical isomers due to the presence of the asymmetric alpha carbon atom. The amino acids contain asymmetric alpha carbon atom. So all the amino acids possess optical isomers due to the presence of asymmetric alpha carbon atom. But some are structurally stable and sterically hindered. They are glycine. Example is glycine. Some amino acids are structurally stable and sterically hindered. Example is glycine. Then the amino acids possess enzymatic activities. Most of the amino acids or proteins possess enzymatic activities. And the amino acids showing colloidal nature and denaturing property. The amino acids exhibit colloidal nature and denaturing property. These are the physical properties of amino acids. Now coming to the chemical properties. First one is decarboxylation. The amino acids will undergo decarboxylation and forming corresponding amines. So the corresponding amines are produced. The amino acids will undergo decarboxylation and forming corresponding amines. The histidine undergo decarboxylation and forming histamine and carbon dioxide. Similarly, tyrosine undergo decarboxylation forming tyramine and carbon dioxide. And also lysine undergo decarboxylation to forming cadaverine and carbon dioxide. Cadaverine and carbon dioxide. So this is the decarboxylation reaction of amino acids. Next is reaction with the alkalis, that is salt formation. Reaction with the alkalis. The carboxyl group of the amino acid relieves a hydrogen ion, H plus ion, and forming carboxylate ions. Forming carboxylate ions. So salt is formed. Here you can see the C from the COOH. This H plus is released and this Na plus bind to this and forming salt or CHNH2COO Na. So this is the reaction with the alkali forming salt. The carboxyl group of the amino acid release a H plus ion and forming a carboxylate ion. This then react with the alkali. Then next one is reaction with alcohol or esterification. The amino acid react with the alcohol to form ester. These esters are volatile in contrast to form the amino acids. The esters are volatile in contrast to the form amino acids. Though here you can see the amino acid react with alcohol, ethyl alcohol, in the presence of the acid catalyst, forming ethyl ester of amino acid. This amino acid react with the ethanol and forming ethyl ester of amino acid in the presence of the acid catalyst. This is the reaction with the alcohol or esterification reaction. Next is reaction with an anhydride. When an amino acid react with an anhydride, anhydride is triketohydranidine hydrate. Triketohydranidine hydrate. This is anhydride. When it react with an amino acid, forming corresponding hydranidine. That is a purple colored complex. So when an amino acid react with an anhydride, forming hydranidine and an aldehyde, carbon dioxide and an ammonia group. This is a reaction with the anhydride. Reaction of the amino acids with the anhydride. Next one is reaction with the dancyl chloride. Dancyl chloride means dimethyl amino naphtha sulfonyl chloride. Dimethyl amino naphtha sulfonyl chloride. When an amino acid reacts with the dancyl chloride reagent, it gives a fluorescent dancyl derivative. So when the amino acid react with the dancyl chloride, forming fluorescent dancyl derivative. This is the reaction with the dancyl chloride. The next one is reaction with acylating agents or acylation. When the amino acid react with the acid chloride and acid anhydride in alkaline medium, it gives thalloid amino acid. So when an amino acid react with the acid chloride and acid anhydride in alkaline medium, forming thalloyl amino acid. Next one is the reaction with the Sanger's reagent. 1-fluoro-2,4-dinitrobenzene is called Sanger's reagent. 1-fluoro-2,4-dinitrobenzene is called Sanger's reagent, FDNB. Here, when the Sanger's reagent react with an alpha amino acid, producing yellow colored derivatives, DNB amino acid, dinitrobenzene derivative of the amino acid. Here this amino acid react with the Sanger's reagent and forming DNB amino acid from yellow colored DNB amino acid and hydrogen fluoride. This is a reaction with the Sanger's reagent. Next one is reaction with the Edman's reagent. Edman's reagent is known as phenyl isothiocyanate. Edman reagent is phenyl isothiocyanate. When an amino acid reacts with Edman's reagent, it gives phenyl thiohydantoic acid, and it finally cyclizes to form phenyl thiohydantoin, Edman's derivative, phenyl thiohydantoin. So here the amino acid react with the phenyl isothiocyanate, that is the Edman reagent, to form phenyl thiohydantoic acid. This then undergoes cyclization to form phenyl thiohydantoin, the Edman derivative. This is the reaction with the Edman's reagent. Here you can see different amino acids. First one is glycine, alanine, valine, leucine, methionine and isoleucine. These all are non-polar aliphatic R group containing amino acids. Non-polar aliphatic R groups containing amino acids. Glycine, alanine, valine, leucine, methionine and isoleucine. Then you can see negatively charged R groups containing amino acids. They are aspartate and glutamate. Negatively charged R groups containing amino acids are aspartate and glutamate. Then next one is aromatic R groups containing amino acids. They are phenylalanine, tyrosine and tryptophan. So the aromatic R groups containing amino acids are phenylalanine, tyrosine and tryptophan. Then positively charged R groups containing amino acids. They are lysine, arginine and histidine. Lysine, arginine and histidine. Then polar uncharged R groups containing amino acids. Serine, threonine, cysteine, proline, asparagine and glutamine. Polar uncharged R groups containing amino acids. Serine, threonine, cysteine, proline, asparagine and glutamine. This is all about classification and properties of the amino acids. Thank you.